Let this serve as a brief introduction to the third deliberation on whether or not eating genetically modified foods is something which we should be doing. This is just a brief outline. You can find more information in the PowerPoint on molecular genetics and biotechnology. In order to understand genetically modified foods and how we make them, we need to do some definitions. First off, what is a gene? Recall that each of our cells has a nucleus, and in that nucleus we have 23, we as humans have 23 pairs of chromosomes. Other organisms, all organisms also have chromosomes, and on those chromosomes are genes contained within the DNA. Here is an image of the X chromosome showing a list of genes that are contained within that X chromosome. In fact, these are just a few of the genes on the X chromosome. The X chromosome is fairly large, so it has over 1,000 genes encoded on it. So genes are small fragments of DNA, relatively small fragments of DNA that are contained on each of our chromosomes. Within those fragments of DNA are specific nucleotide sequences. A rough definition of a gene is a sequence of deoxyribonucleotides that code for a product. Those products are often proteins. The way that genes are used is that when we want to create a product from a gene, we pair ribonucleotides with an uncoiled DNA sequence. And the nucleotides that we pair with those DNA deoxyribonucleotides are of the RNA variety. Those ribonucleotides are assembled in a line to create an RNA transcript. That RNA transcript is shuttled to the cytoplasm, where it is bound by a ribosome that will translate it. The process of translation necessitates tRNAs that will pair with the RNA in the cytoplasm. Each of the tRNAs will bring in an amino acid, and the ribosome will catalyze a chemical reaction between those amino acids to create a growing amino acid chain. That is the protein. So the central dogma of molecular biology is that we read the DNA, convert it to RNA, and then we translate that RNA into protein. We can edit the DNA in organisms, and that is because all of the DNA in all of the organisms has the same structure. Different sequences, but the same structure. We can use enzymes to cut out segments of the DNA and replace those segments of DNA with fragments that contain the desired sequence being one that codes for something we want in an organism. For example, sweetness in fruits or vegetables. Here are some examples of genetically modified foods. Did you know that some of the rice, tomatoes, corn, and salmon that you eat is genetically modified? In this table, you can see how those foods are modified. Some potential advantages for genetically modifying foods is it's possible to create healthier foods with greater nutritional value. Sometimes we can create foods without allergens, and with respect to tomatoes specifically, it makes them easier to transport and have a longer shelf life. Another advantage is that we can generate more product per area of land, or in the case of salmon, more product in a given period of time. This graphic shows some other advantages. Not only does it increase our crop productivity, but it also helps to conserve biodiversity. It reduces agriculture's footprint, and it helps mitigate climate change. I'll leave it to each of you to investigate these claims, as this graphic was put together by a genetically modified food organization. There are some potential disadvantages. I won't read these to you, but I'll also have you consider those as you enter your deliberation. Again, you are meant to determine whether or not you personally should be eating genetically modified foods.